Hello again, my dear students. We are moving to a new topic about gene mutations and how they are detected. If you are still able to remember my discussion about genetic variations and mutations, I mentioned that there are a number of ways that a certain part of the genetic material can be altered. Some of these alterations, especially those that change the amino acid sequence of DNA, or those that result in premature termination of the replication process, would often lead to phenotypic changes. These phenotypic changes may be irrelevant and some may be pathologic. In this topic, the techniques and how to detect mutations in a clinical setting will be taught. There are basically two major ways on how mutations are detected. One indirect and the other is direct. Biochemical methods of mutation detection are a group of methods that indirectly determine the presence of mutation. Indirect because they don't directly examine the nucleic acid composition of the gene. Instead, biochemical methods detect the altered protein products as a result of the genetic mutation. Any genotypic change in the genetic composition of a certain locus may often result in phenotypic changes which include a change in protein structure or function and ultimately lead to the production of altered protein products. Rather than searching for potential point mutations in the gene, biochemical methods analyze the change in protein structure or function. These are detected using biochemical methods. There are three most commonly used biochemical methods to detect the presence of mutation. These are enzyme immunoassays, immunohistochemistry, and automated methods. The first biochemical method to indirectly detect gene mutations is enzyme immunoassays. This technique makes use of specific antibodies or other ligands that detect the presence of altered protein products. And aside from antibodies, this technique also involves the use of enzymes and substrates that are important for signal detection and quantification. Enzyme immunoassays are flexible and can be used for high-throughput screening, especially when the reaction is allowed to happen in a 96 or 192 well plate matrix. Enzyme immunoassays allow for the detection both for antigens and antibodies. In this example is an enzyme immunoassay format used to directly detect antigens. Antibodies that are specific to the target antigen are immobilized in a solid matrix such as well plates. Then, the sample containing the antigen is added to the well for antigen-antibody binding. Then, the secondary antibodies or detection antibodies conjugated to an enzyme such as horseradish peroxidase or alkaline phosphatase is added to the wells. The substrates are then added that will result in color development. The intensity of the color is proportional to the concentration of the target. Immunohistochemistry is also another biochemical method to quantify altered protein products as a result of mutations. This technique, just like enzyme immunoassays, still makes use of antibodies or ligands to detect protein and or protein product epitopes. But instead of having the reaction to proceed in a well, the reaction is performed on thin slices of either fixed or frozen tissue samples. Immunohistochemistry follows the following steps. For paraffin-embedded tissue samples, the first step is to deparaffinize the tissue, followed by a rehydration process. The deparaffinization step is omitted when handling fresh or frozen tissue samples. Then, antigen retrieval proceeds. Antigen retrieval is a process that will expose the epitopes or the antibody binding sites of the tissue. This step is especially useful for paraffin-embedded tissues because paraffin in routine tissue processing tends to obscure the epitopes. After antigen retrieval, blocking with either hydrogen peroxide, UV light, or 1% serum happens. Blocking is essential for preventing background signals from non-specific binding of antibodies or other reagents to the tissue. Even if the antibody has high specificity towards the target, intermolecular forces can promote non-specific binding to other molecules. That's why the blocking step is important. And then, after blocking, the primary antibody specific for the antigenic target is added, followed by the addition of the secondary antibody conjugated with an enzyme. The substrate is added for color development after. The tissue slide is subjected to imaging or microscopic examination for the detection, localization, and quantification of the target. As mentioned, imaging of the tissue in immunohistochemistry will provide the investigator an idea if the antigenic target is successfully detected. Once detection of the target is successful, imaging will also give an idea where the target is localized in the tissue. Immunohistochemistry can also help in the quantification of the antigenic target with the help of imaging softwares. That is why immunohistochemistry really is an important technique for the indirect detection of mutation, as it allows the detection of protein abnormalities in situ. Thus, the tissue and intracellular location of mutant proteins, or the lack thereof, can be observed. The next biochemical technique is high-performance liquid chromatography, or HPLC, which has been adapted for automation. The separation principle of HPLC is based on the distribution of the analyte or the sample between a mobile phase, which is composed of an eluate or the solvent, and a stationary phase, which is the HPLC column. When a test sample is dissolved in the mobile phase, it is introduced to the HPLC by injection with a syringe through an injection port in the column. Pumps will force the sample through the column, and depending on the chemical structure of the analyte, the molecules are captured while passing the stationary phase. Elutions are monitored by a detector that produces a readout or signal peaks as the sample components elute. Another automated method for the indirect detection of gene mutations is gas chromatography. This technique involves the separation of a vaporized sample through a column of inert carrier gas, or the mobile phase, and a high boiling point liquid that differentially absorbs molecules, or the stationary phase. The strength of the interaction or dissolution of the sample components into the liquid phase will result in varying retention times in the column. The effluent from the column is monitored by a detector, such as a flame ionization detector that is sensitive to organic molecules. Mass spectrometry is also an indirect method to detect genetic mutations. A mass spectrometer converts molecules to ions that can be moved in a magnetic field based on their charge and mass. In this automated method, an ion source sends high-energy electrons that hit the target sample molecules, separating them into ions, usually cations with a loss of 1 or 2 electrons. The ions themselves may further fragment into smaller ions and neutral particles. This collection of particles is accelerated and focused into a beam through a magnetic field that detects or deflects the ions according to their mass and charge. The readout of the instrument is a spectrum with a mass over charge value on the x-axis and the abundance of the ion on the y-axis. Molecules can therefore be identified by their characteristic spectrum or set of peaks. Now we move on to the next major set of mutation detection techniques. This time, instead of quantifying or detecting the protein products resulting from gene mutation, nucleic acid analyses directly look into the composition of specific nucleic acid fragments to check for mutations or single base pair changes. Biochemical detection and/or characterization of proteins using the biochemical methods just described may be part of a clinical chemistry laboratory menu. Mutation detection by analysis of nucleic acids is considered the classical molecular methodology. Although it is the most definitive method for detecting mutations, DNA sequencing may not be appropriate, especially for high-throughput procedures. A number of techniques have been designed for the detection of DNA mutations, from single base pair changes to large chromosomal rearrangements, without having to determine the primary DNA sequence. That is why we have these nucleic acid analytical techniques for gene mutation detection. The first nucleic acid-based analytical technique for the detection of gene mutations that I will talk about is single-strand conformation polymorphism, or SSCP. Single-strand conformational polymorphism analysis is a simple and sensitive technique for mutation detection and genotyping. The principle of SSCP analysis is based on the fact that single-stranded DNA has a defined conformation. Altered conformation due to a single base change in the sequence can cause the single-stranded DNA to migrate differently under non-denaturing electrophoresis conditions. Therefore, wild-type and mutant DNA samples display different band patterns. SSCP analysis involves the following 4 steps. First is polymerase chain reaction amplification of the DNA sequence of interest. Second is the denaturation of the double-stranded PCR products. After the denaturation process, the third step is cooling of the denatured DNA or single-stranded DNA to maximize self-annealing. And lastly is the detection of mobility difference of the single-stranded DNA by electrophoresis under non-denaturing conditions. Several methods have been developed to visualize the SSCP mobility shifts. These include the incorporation of radioisotope labeling, silver staining, fluorescent dye-labeled PCR primers, and more recently, capillary-based electrophoresis. This is an example of a silver staining or SSCP analysis for the detection of a threonine to methionine mutation at the 594th amino acid position. The TM lane reflects the presence of the heterozygous mutant genotype. The TT lane shows the homozygous T wild-type genotype or the normal genotype, while the MM lane shows how the homozygous mutant genotype looks like. This figure further suggests that a change in the sequence of amino acids from a mutation will definitely show different electrophoretic band patterns of the DNA. Allele-specific oligonucleotide hybridization is also a method for testing known mutations. This makes use of an allele-specific oligonucleotide or ASO, which is a short piece of synthetic DNA complementary to the sequence of a variable target DNA. It acts as a probe for the presence of the target in a southern blot assay, or more commonly in the simpler dot blot assay. In this method, DNA products generated by polymerase chain reaction are dotted in duplicate membranes. One membrane is dotted or hybridized with an oligonucleotide corresponding to the wild-type sequence, and the other to a mutant sequence. Normal individuals show hybridization only with a normal probe. Homozygous individuals show hybridization only with a mutant probe, and heterozygous individuals hybridize with both probes, so both the normal and the wild-type probes. In specific annealing temperatures and conditions in allele-specific hybridization, the allele-specific oligonucleotide acting as a probe will not bind to a near complementary target sequence with one or two mismatched bases, whereas a probe with a perfect complementary sequence will bind. This is the basis for the detection of mutation using the ASO hybridization technique. The next nucleic acid-based analytical technique for the detection of gene mutations is restriction fragment length polymorphism, which has an acronym of RFLP pronounced as RIFLIP. It is a commonly employed tool to detect the small but specific variations in a sequence of double-stranded DNA. It is based on the specificity of restriction endonucleases, which recognize a set of nucleotides called restriction site and cleave the DNA at those sites. A specific RIFLIP pattern emerges on electrophoretic separation of digested DNA, producing variable lengths of cleavage fragments which are characteristic of a sequence of DNA. Individuals, species, or organisms can be distinguished on the basis of their RIFLIP pattern. RIFLIP are widely used in genotyping, DNA fingerprinting, mapping of genes, and diagnosis of genetic disorders. The typical workflow of restriction fragment length polymorphism involves restriction digestion, fragment separation, southern blotting, probe hybridization, and visualization. In the first step, purified genomic DNA is digested with one or more restriction enzymes. The choice of restriction enzymes is usually based on the ability to distinguish genetic variability and the cost of the enzymes. The digested fragments are then separated by gel electrophoresis and should appear as a continuous smear on the gel due to the broad distribution of fragment sizes generated by the enzymes. To detect the desired fragments, the gel-separated DNA fragments are transferred to a nitrocellulose membrane for handling and detection. A label single-stranded DNA probe is hybridized to the membrane to identify a subset of fragments. The results are visualized to reveal the unique RIFLIP fingerprints. Another nucleic acid analytical technique is allelic discrimination with fluorogenic probes. This technique is only applicable for assays that use thermal cyclers with fluorescent detection support, like the real-time PCR thermal cyclers. This methodology employs two fluorescently labeled probes that are complementary to either the normal or mutant sequence, and the presence of the corresponding fluorescent signal indicates whether the sequence is normal or mutant. The two most commonly used fluorescent dyes to label the probes are FAM and VIC. Say, for example, the sequence specific to the normal target sequence is labeled with FAM, and the sequence specific for the mutant sequence is labeled with VIC. An increase in FAM signal indicates a normal sequence, and high VIC indicates a mutant sequence. If both fluorescent dyes are detected, the test sequence is heterozygous. This technique is currently used to genotype single nucleotide polymorphisms. If you want to do additional readings, you may always refer to the following references. This concludes my discussion about gene mutation detection methods. Remember that there are two main technique classifications: biochemical methods and nucleic acid analyses. Thank you for watching this video and have a good day, everyone.